Hello, and a very warm welcome to today's Nucleic Acid Insights webinar titled Assessing mRNA Stability and Integrity Using Mass Photometry. I'm Alison Gash, an editor at BioInsights, and joining me today is Jacintha Victoria, who will discuss present quantitative results from inhibitor binding studies using mass photometry. After the presentation, we'll have a live Q&A session, so please feel free to pose your questions to our speaker using the Ask the Question box at the bottom of your screen, and we'll try to get to them during the session. I'd also like to draw your attention to the resources on the right-hand side of the screen, where you can find more information on the topics covered today. Now, I'd like to briefly introduce our presenter. Jacintha Victoria is a strategic marketing and analytical leader with over a decade of experience in the biotechnology and gene therapy sectors. As the Market Development Manager at Refeyn Oxford, she leads initiatives and campaigns to drive the adoption of mass photometry across critical applications in cell and gene therapy, including AAV, LVV, adenovirus, and RNA-based platforms. With a deep understanding of the emerging analytical technologies, Jacintha advocates for mass photometry as a transformative tool to enhance viral vector characterization, accelerate product development, and strengthen regulatory preparedness. So, without further ado, I'll hand over to Jacintha to kick off today's presentation.
Thank you, Alison. Good day, all. As introduced, I'm Jacintha Victoria, Market Development Manager for the CGT space with Refeyn. And for today's presentation, I would like to talk about assessing key quality attributes for mRNA product using mass photometry. So in today's presentation, the key things that I would like to talk about are the key questions that you would ask yourself during characterization of an RNA drug product and how some of those questions can be answered using our technology, which is the mass photometry. And finally, having benchmarking data of mass photometry against orthogonal methods in the industry, just to draw evidence for the reliability and accuracy of the measurement. So to start off with the structure of mRNA, so there are a number of components that make up your mature mRNA molecule, and each of it has a significant functional activity. So we have the 5' end, which has the cap structure. That's nothing but methylated guanosine, which follows through with triphosphate linkage. So together, they help protect the mRNA from degradation. On the other side, which is the 3' end, you have the poly-A tail, which is a sequence of 150 to 250 adenine residues. And the main role it plays for mRNA is to maintain stability and to enhance translation efficiency. In the center, you have your coding sequence. So this coding sequence encodes for your gene of interest, and any chemical modifications on your coding sequence help enhance the stability with minimizing your immunogenicity when administered to patients. And then finally, you have your 5' UTR and 3' UTR, which are your untranslated regions flanking your coding sequence. And the main role it plays, again, is to enhance your translation efficiency and to maintain stability. So this table here summarizes the critical quality attributes of your mRNA product as defined by regulatory agencies. So going from left to right, you have your quality, you have the attributes that fall under each of those qualities, and finally, you have the methods that help measure the attributes in the table. So the attributes that are highlighted within the orange box are the ones that are currently can be assessed by mass photometry. So one thing to highlight here is, although the methods mentioned here in this table are very widely used in the industry for measuring different attributes for mRNA product, they broadly come with a number of limitations. They require high turnaround time, is quite complex to run, has high cost limitations that go with it. And finally, the major impact is because some of these methods require your sample to be modified during the measurement process, it can really interpret or cause a skew in the interpretation of the results. So as I touched on before, when you look at biophysical characterization of a molecule, especially RNA, there are some key questions that you look at. So I've broadly classified them or grouped them into 3 categories. So you have the analysis of complex formation. So your RNA molecule could interact with or bind with other molecules to form a complex. So as part of this, you're looking at what those interacting species are, the functionality of the species, and more importantly, by forming these complexes, how does it impact the potency of your final products? Under the interaction studies, you're looking at the interaction between the different molecules, your RNA molecule with other proteins, DNA, and RNA. So here you're looking at the stoichiometry of the complex formation and the strength of the interactions. Again, keeping in mind how that impacts the potency or efficacy of your drug. And finally, you have the sample characterization, where you're mainly focusing on the attributes themselves. So you're looking at purity of your product, quality of the product, and mainly the integrity of the product, which during the process development phase could be impacted by the formulation studies, where you're changing different formulations to understand which is best for your product, and also during your process optimization phase, where you're optimizing your process and product to get the best output with no impact on the yield and no impact on the quality of the product. So with keeping these questions in mind, I wanted to focus on how these questions fit into the different stages of a drug development process. So you have from left to right, you start off with the research and discovery phase, then you move your drug into the process development where, like we previously touched on, you develop on the process itself to get better yield and high quality. And finally, you look at the quality of the product. So in the research and discovery phase, you're mainly focused on screening multiple molecules and trying to understand the interactions and behaviors that come out of it. So mainly speaking here, you're looking at, again, complex formation, as in your molecules interacting with other molecules, the functionality of your final product, the stoichiometry of it, and the strength of the interaction. So once you have products that look ideal for you with the desired outcome, with no impact on potency and efficacy, you now move them into the process development phase. So in the process development phase, again, you're looking at your formulation study, you're looking at changes in the optimization of your process, which could have an impact on the integrity or stability of your product itself. Again, here, through all of this, you're looking at, yes, your purity, your quality, with no compromise on the efficacy of the product. And finally, now it moves into the quality control phase, where you're mainly focusing on the attributes itself. So here, you've passed your optimization, you've passed your initial screening. So here, you're mainly looking at the purity of your final product, what are the impurities present, the quality of the product, which also includes the efficacy. So one thing to highlight here is the efficacy of the product or the potency of the product is one key attribute that is monitored throughout your drug development process. So although some of these questions are already answered using the methods that we discussed in the table, that's widely used in the industry, there are some limitations that were highlighted. So what does an ideal tool look like then? So your ideal tool should give you accurate measurement, should do it in a faster, easier, and a cheaper fashion, and all with native measurement, because your native measurements will give you more reliable data than when you do it in non-denatured or denatured conditions. We'll talk about the pros and cons of that in just a minute. So now I would like to introduce a new technology, which is the mass photometry, because it checks all of the requirements needed for an ideal tool to measure RNA molecules. So as the name goes, mass photometry measures your mass of your individual molecules in solution using light. So going from left to right again, this technology is nothing but a glass slide which has your particles in solution. Now, when illuminated with a laser, the particles scatter light. As you can see in the second column, you have a contrast image that is formed from each of these particles, which look like circular dots. Now, these contrast dots are actually proportional to the mass or density of the particle. So darker the contrast, heavier the particle. Your contrast now linearly scales with the mass of these particles. So what the software now does is, it takes the contrast information from every single molecule that's measured and converts it into mass using a mass calibrant. In this case specifically, we use mass Ference P1. And finally, you have the output of the mass versus counts, which is a mass histogram. So here you're able to understand the different species or the complexes formed with your molecule in your sample, all differentiated based on mass. So 2MP is a mass photometer that has been specifically designed for proteins, antibodies, and nucleic acid measurements. So like we previously touched on, yes, it can answer some of the questions under each of these categories, the analysis of complexes, interaction studies, and finally the sample characterization. But broadly, it can also look at your low abundant species, which cannot be measured on other methods, heterogeneity in your samples, like the different oligomeric states which define the purity of your sample. And very key is it can be used as a process monitoring tool, which will show evidence in just a bit. So overall, your 2MP, which is a mass photometer, is a very quick tool, and it can do label-free measurements, all of single molecules in their native state, all within minutes with very low sample volumes. So now let's go into how 2MP is actually able to answer some of these questions. So we'll start off with sample characterization, where we look at the integrity of the molecule, the purity as in the purity profile, where you're looking at different oligomeric states, and also other impurities measured, other impurities that are co-purified with your product. So first thing here, this experiment was done internally on the 2MP, where we looked at different mRNA molecules and saRNA molecule, where on the right of each of the histogram, you're looking at the expected length of each of these RNA molecules defined on an orthogonal method. And each of these histograms are done in replicate, so that just adds more evidence to the reproducibility or reliability of measurement from the mass photometer. As you can see with all of these molecules, the measured length of your mRNA or the RNA molecule against the expected very closely aligns. And that's further evident from the correlation fit graph on the right, where you have the best correlation fit between your expected length of your RNA versus the measured length of your RNA, thus helping us conclude that 2MP can reliably measure the size of your particles, which helps us assess the integrity of your mRNA molecules in your product. So the next thing we looked at was to understand the purity profile of our mRNA product using mass photometer. So very widely speaking, there are two methods that's used in the industry to measure mRNA purity. Those are your capillary electrophoresis and your high-performance liquid chromatography. But both of these methods measure the purity of your mRNA product in reducing and denaturing conditions, whereas mass photometry does it in its native form. So this data here was generated again internally on a 2MP, where we took the mRNA EGFP molecule and measured on 2MP in its native buffer conditions, on the left called non-denaturing, and on the right under denaturing conditions, where it was reduced using urea and heated to denature the molecule. So just looking at the image visually, you can see, although it was the same molecule measured, you can see a difference in the purity profile between the non-denatured and the denatured. So the non-denatured, you have an abundance of your monomers, which is roughly around 980 bases, followed by smaller populations of your other oligomeric states, which are your dimers and your trimers. Now, looking at your denatured sample that was measured on 2MP, you still have an abundance of your monomers, but you have negligible to nothing of your other oligomeric states present. So one thing to highlight here, as you might have noticed, there's more abundance of your monomers in the denatured versus the non-denatured. And that's because of your reduction and denaturation that breaks down your other oligomeric states into your monomeric state. So why is this important to show this comparison of non-denaturing condition versus denaturing condition? So as you know, like I mentioned, most of these methods that are available in the industry use denaturing conditions. So if that was so, you are actually misinterpreting the purity profile of your RNA product. Thus, again, helping us show that 2MP is very reliable because it measures your RNA in its native state. This gives you the accurate or the actual purity profile of your mRNA product with no skew to the interpretation. And then finally, so this is a data snippet from a publication done by Rick Biologics, where they looked at dsRNA impurity. So as we all know, in the mRNA production process, there is a step called IVT, which is in vitro transcription. So during this step, your mRNA is produced, but together with that, you have an impurity generated, which is your dsRNA. It is an unwanted impurity because of its immunogenic responses when administered to patients. So this lab wanted to understand if mass photometry can be adapted to understand the presence and quantification of these impurities. So on the right, you see the image going down where they measure the individual components. So we have the A image where they've measured the dsRNA separately on the MP, Then they followed through with the antibody. So the J2 antibody is specific to your RNA. And then finally, the C is where they've combined the two and done the measurements. So as you can see from the mass differences, you have the last two populations that are coming up, which shows you the presence of your dsRNA, but the shift in populations comes from the different stoichiometries, thus helping us conclude that mass photometry yet can also be used to measure another impurity in your mRNA product, which is dsRNA. So the next thing I wanted to discuss was analysis of complex formations. So this data is obtained from a publication, so where they've looked at oligomeric states or stoichiometries of your miniature type VF CRISPR-Cas nuclease. So as we know, Cas12f is gaining more interest in the industry. It's a more compact version of Cas9. And because of the guide RNA direction, it gives you more targeted genome editing, and that's why it's gaining more interest. So in this experiment here, they initially looked at the different components. So your different components are your SP-Cas12f, your guide RNA, and finally your double-stranded DNA. So they looked at each of those separately. So they first looked at the yellow represents your Cas, which is mostly in its monomeric form without the guide RNA. And then the guide RNA, which was measured separately. And finally, you have your dsDNA. So once mixed together, so once they mixed your Cas together with gRNA, it was able to form a complex. So your Cas12f forms a homodimer with a gRNA. And you can see the shift, which is represented by the molecular weight measured. And then when you add the dsDNA, it forms a ternary complex. Now, the data is not shown here, but this was confirmed with the cryo-EM structure, which was measured separately, thus indicating that your catalytically competent Cas12f complexes usually contain two subunits of your Cas nuclease, one of your gRNA molecule, and one of your DNA molecule. And finally, the last section I would like to cover is the interaction studies. So under interaction studies, we wanted to look at quantifying the inhibitor potency for small molecule binding. So in this case, we wanted to screen three different small molecules, your X1, X2, and X3, to promote and stabilize formation of 16-mer complexes. So 16-mer complexes are mostly in its inhibitory form. And as you can see, with increase in concentrations of your X1 and X2 molecules, you see an increase in formation of your 16-mer complex, as also evident on the histogram shown on the right. Whereas with small molecule X3, when tested at varying concentrations, showed no impact. Again, also evident on the histogram on the right, where you don't see a complex 16-mer complex formation. So again, this helped us understand that MP can play a very critical role, a crucial role, to understand these structural changes which are induced by small molecules. So now that we've shown that MP is very reliable, it can help you measure the integrity of your RNA molecule, your intact purity profile in its native state, and also try to understand the complex formations for Cas9 and also for your small molecule interactions. Now we wanted to show you how reliable that measurement is by comparing it to orthogonal methods in the industry. So this is a data snippet from a publication done by Pfizer, where they did measure a number of RNA molecules, as shown in the table here, with known length and known mass on both the charge detection mass spectrometry, again, very widely used in the industry, against mass photometry. So the data shown below, the histogram of your RNA molecule, is only a representation of one of the data sets. But as you can see, the profile very closely aligned between the two methods. Now, taking all of the information, especially the mRNA length obtained from both of those methods, CDMS and MP, for all of the 9 molecules in the table, we put a graph together to understand the percentage error, as in the error rate of the measured mRNA length on each of those methods against the expected. So blue bar here represents CDMS, and the orange bar represents mass photometry. As you can see, all of the data obtained for mass photometry, the error rate was well within 2%, as in there was only a 2% deviation between the measured and the expected. Again, thus helping us conclude that mass photometry is reliable and accurate for mRNA measurements as its industrial counterparts. So one of the requirements for being an ideal tool is also process monitoring. So here we wanted to look at, or Sanofi looked at, understanding if any changes in process could impact the measurements done on mass photometry. So again, this is a publication done by Sanofi where they took the mRNA post its capping and tailing phase and they used it during process optimization. So as in any product optimization or process optimization, you have changes in your buffer conditions, changes in salt concentration, and inclusion and exclusion of surfactants. So what they did here was they took the same mRNA molecule and put it through different buffer conditions and then measured it on the MP. As you can see, the error rate between all of the measurements for the 3 samples that were done here was well within 3%, thus helping us conclude that mass photometry is reliable and reproducible for the measurements of samples in any number of matrices, thus showing that NP is not really impacted by matrix effect, thus helping us conclude that MP is an ideal tool for process monitoring for mRNA production. And then finally, this is a data snippet from a publication by Genentech, where they looked at understanding if MP can be adapted as a stability monitoring tool for RNA. So here they took mRNA, and this is the control, as in the non-treated, which is the right top, where you can see 2 different oligomeric states. So you have your monomers, which is in abundance, and then you have your non-covalent dimers. So one thing to highlight here in the paper, they have also done the same measurement, the control measurement, on 2 other methods, which is IPR-PLC and CGE, which uses reducing and denaturing conditions. So both of those methods did not pick up the non-covalent dimers, which were actually observed on the MP. Again, thus highlighting MP is very reliable to measure the actual intact populations in your sample. So they took the mRNA, and for the heat stress, they heated it at 37 degrees for 5 minutes. And once heat stressed and tested on the mass photometry, you can really see a shift in the population. So the dimeric states diminished, and you could really see an increase in the peak region by about 34%. Thus, again, helping us conclude that mass photometry is reliable, can be used as an in-process monitoring tool, and can also be used for stability monitoring of RNA products. So as we know, RNA, nucleic acid, is negatively charged. So as part of the mass photometry measurement, there is a cover slide that's used as a consumable for measurement. So this is an experiment we did internally to try and understand if any modification to the surface is essential or has a significant impact on the measurement. So the image on the histogram on the left shows you 2 different glass slides that were used. One is the untreated glass slide highlighted in blue, and the other is the coated glass slide, which is nothing but a PLL, polyl lysine, which is a cationic coating, done in bluish gray. As you can see, when measured on untreated glass, the ssRNA ladder did not bind, therefore no measurements were recorded on the MP. Whereas on the cationic coated slide, we were able to accurately measure the different populations, the abundance of the populations within the ladder material. Thus helping us highlight that cationic coating, such as the PLL, is very very much required for nucleic acid measurements, which is also highlighted in the protocols published by Refeyn. So one thing to mention here is that Refeyn is actually working on releasing a ready-to-use slide for NA or nucleic acid analysis. So stay tuned with the Refeyn network to know when we'll be releasing that slides for nucleic acids. So this is the specification for the instrument. So one thing I would like to highlight here on the right, you have a full mass range. So you're going from 30 kilodalton to 5 megadalton. And as you can see, because of the wide mass range that the 2MP can cover, you're able to measure very different molecules and vectors, both viral and non-viral, on the instrument. And finally, to introduce a portfolio. So we have 3 products on our portfolio. So we have 2 mass photometers, which is the 2MP, which we just spoke about, which is mainly designed for proteins, antibodies, and nucleic acids, covering a mass range of about 30 kilodalton to 5 megadalton. We also have the SAMEX, which is also a up version of the 2MP, which has been designed for AAV characterization. And that covers a different size range, covering the sensitivity of the AAV vectors, and that's about 500 kilodalton to 6 megadalton. Both of these MPs, which is the 2MP and the SAMX MP, have the manual and automated workflows. And the SAMX MP with its GMP software release can be adapted in both PD and GMP environments. And finally, we have the newest inclusion to our portfolio, which is the Queretro MP, which is also called the macro mass photometer. So that's called MMP mainly because it not only does mass measurements, but it also does size. And this has been designed to target larger viral vectors like adenovirus, LVVs, LNPs, and they cover a size range of 40 to 150 nanometer. That concludes my presentation. Thank you for listening in, and I'm happy to answer any questions.